a long day yesterday, I would like to ask you to put your cell phones on the silent mode. The Institute of Technology and Immunobiological's Bio-Manguinhos from the Oswald Cruz Foundation has the pleasure of welcoming you at the 8th International Symposium on Immunobiologicals 2024. The goal of the event is to integrate scientists, some researchers, and institutions towards the development and production of immunobiologicals, in addition to providing visibility and providing capacity building to professionals and promoting also opportunities and partnerships in the biotechnology sector, stimulating an environment of scientific development and technological growth at the national and international level. We would like to thank the following sponsors and supporters: Sanofi, Promega, GSK, Biotec, Sativa, Sykes, Cepheid, Beckman Coulter, Bionovis, AstraZeneca, Getúlio Vargas Foundation, and Croda. We would like to recommend that you gain access to the application of the event directly through our website or through the area of participants that is being shown on the screen. Through this application, you're going to be able to ask questions directly to the speaker and to participate in the set of customer surveys, including a professional profile, so that all participants at the event will get to know each other, exchanging information and having access to all the works that have been enrolled. And I would like to recall that our event is taking place in hybrid fashion, and you can follow the whole program live through our application. You can also participate asking your questions here in the audience or online through the chat room, and we'll make sure it will get to the chair of the panel. The public here in the audience have simultaneous translation equipment, and for the online audience, all you have to do is click on the language you prefer on the icon in the lower portion of the video. Our lectures will be recorded in our application. You can listen to them at your whenever you feel like it. We've had a campaign, our friends in the south of Brazil, and 1 kilo of non-perishable foodstuffs or feed for animals, hygiene items, cleaning items. You can bring them. If you haven't brought these items, there is a little market. There's a Sugarloaf market right next to us. I think it's very important for us to undertake this joint action, and you can leave your donation at the tent we have right next door. Now, starting the presentations of posters, I'd like to welcome on stage Maísa Oliveira Ribeiro. She will present the topic Respiratory Virus Panel and Respiratory Virus Panel 2, Bio-Manguinhos Molecular Typing Assay for discriminatory diagnosis of respiratory infection. Please. Good morning. Do you all hear me? Well, let's go. My name is Marisa. Good morning. I'm a biologist. I work at the molecular diagnosis laboratory in Manguinhos. We develop kits. I'm going to present the respiratory virus panel 1 and 2. It's a molecular typology tool that is a discriminatory diagnosis tool for respiratory diseases, the main respiratory viruses, which have a clinical importance for Brazil and immunological importance for Brazil, and are the target of molecular diagnostics on this panel are SARS-CoV-2, S2, which is the which caused the latest coronavirus pandemic, influenza A and B viruses that lead to seasonal respiratory illnesses, the metavirus, human metavirus, which leads to common flus, the respiratory virus that is present and circulating among our population, which may lead to severe bronchiolitis in young children and may lead to death as well. The rhinovirus, which is commonly circulating in Brazil, and it's very characteristic of hospitalized kids. It's very important because it affects children at the hospital, and that may lead to more severe diseases than the adenovirus, which is present throughout the year. It's not seasonal. It causes common cold and flus, but it's present throughout the year in all seasons. As for clinical aspects, I can tell you that all these viruses co-circulate in the population and they present similar symptoms. Just from the clinical diagnostic, we can discriminate what's the cause, the virus that causes the respiratory disease, and people can deal with the infections easily without the need for specialized clinical assistance. But these viruses can lead at times to severe illnesses that may lead to death. And that's very worrisome among children, pregnant women, the elderly, and health professionals. As for prevention, what the WHO recommends is personal hygiene, avoiding contact with people with symptoms to avoid contamination, and prevention through vaccines against SARS-CoV and seasonal flus. As for diagnostics, Bio-Manguinhos, which is the arm of Fiocruz that develops molecular kits and vaccines, has quick tests, but it has acted also in the development and standardization and distribution of molecular tests for respiratory viruses that has intensified because of the pandemic. We had to develop, molecular tests to detect mainly SARS-CoV. And this latest test was developed on the left as a molecular test that discriminatory test for influenza A and B and SARS-CoV-2. This test is being distributed to breastfeeding women. A lot of the patients that had respiratory symptoms and had undetectable results for MB, AB, and SARS-CoV-2, we noticed that they have symptoms that may be caused by another virus. And at this point, we saw the need to develop a molecular panel, discriminatory molecular panel that would be more embracing, encompassing, and that could take care of other viruses that could lead to the aggravation of other illnesses, FB, AB, and SARS-CoV-2. We've had a long-term partnership with a benchmark laboratory at IOC led by Dr. Maria Siqueira and Dr. Fernanda Mata, and we have listed the viruses that would be most prevalent among the population. We've listed the viruses that would make up a new panel for respiratory diseases. We have two multiplex reactions. One of the BR1 reaction detects influenza A and B, pneumovirus, human pneumovirus, and then adenovirus, respiratory illness, rhinovirus. And these two reactions that take place at the same time have the internal control, the room RSP. These are respiratory reactions, discrete and sensitive. They're part of a multi-centric study. We have distributed these reactions over 100,000 reactions as a whole sum of research. It has not been registered at Anvisa. These reactions have been developed by the laboratories around Brazil. We have had the chance to develop a cooperative study with a laboratory at the Medical School of São Paulo under the leadership of Dr. Mauricio Nogueira. This group has a great deal of expertise in the development and production of vaccines against arboviruses. In this cooperative study, we had access to 48 symptomatic patients that had been previously tested in-house through the CDC protocol at the medical school for SARS-CoV-2. This SARS-CoV-2, these patients, 10 of them had positive results for SARS-CoV-2. When they were tested with the in-house method, the same 48 patients were then tested for VR1, VR2, which is the Bio-Manguinhos test. And what happened was we had 100% agreement, and the results of positive results of 10 positive samples from on the in-house test were positive from VR1, VR2. Of the 10 positive samples, there were 38 samples that were not detected for SARS-CoV-2 through the medical school test. Of these 32 samples, 11 were detected for other respiratory viruses when they were tested for a VR1, VR2 panels. Now, with this, through this investigation with VR1, VR2, they were positive for other viruses. They were distributed. Of these 11 other samples, 2 were positive for influenza A. One of them was positive for metapneumovirus, human pneumovirus, and then RSV, 3 for rhinovirus, and 3 for adenovirus. And to our surprise, another sample was positive, triple infected. Among these positive samples, one of them was positive for SARS-CoV-2, COVID-2, and RSV at the same time, concomitantly. This is important because we know from the literature that co-infected patients may have the aggravation of the disease. It was important to know what when it was amplified, we detected also for other respiratory viruses. This sample, it was a 9-month-old child with moderate symptoms of a respiratory syndrome. Here we can notice the amplification curves of the 3 viruses and their respective CTs through the PCR, real-time PCR test. Those Winteri curves that leave no doubt as for the real diagnostic, and that was important for us to be able to visualize and know that these results were discriminatory and definitive because the 3 viruses had been clearly identified. We did a sample later. The week we were present at the medical school, we got the epidemiological report from the region, and we ascertained that at the time we were doing the cooperative test at the medical school, the most prevalent viruses in that town were SARS-CoV-2 and also rhinovirus. And we can notice also that the age bracket that was most affected was children under 12 years of age, under 12 years of age. We can identify a triple infection in a child under 12 years of age from the viruses most prevalent in the town. These are the data from April. They tested 450 samples from the in-house test, and they were not detected for SARS-CoV-2. These undetected samples were tested on our syndrome panel, and they had only been tested for SARS-CoV, and they were positive for 335 samples, and the other respiratory viruses that are part of our syndrome panel were detected here. And see in the graph, in the bar graph, among these samples, the most prevalent one was RSV. As for the conclusions, we can see that the panel was extremely capable of detecting and discriminating respiratory viruses that cause respiratory infections that are most prevalent in Brazil. We have observed also that it is able to discriminate the virus that's causing specifically that syndrome, and it may discriminate also what we know that some are more severe than others depending on the age bracket and the patient conditions. That's very important to our clinical studies. It's important to from a laboratory perspective. It's important because it's 2 reactions that detect 7 viruses all at once. You don't have to do it in-house individually. You can detect it through the syndrome panel. And we also have the expectation that this panel will be certified at Anvisa by the first semester of this year. We'll be submitting the dossier of certification by the middle of the year. And once it has been certified, it will be a very important clinical tool and to detect epidemiological infectious diseases. I'd like to thank AMBR, the Medical School of São José do Rio Preto, which was very helpful in our tests, and the smallpox and mumps laboratory of respiratory viruses of IAC. Thank you. Thank you, Marisa, and congratulations on your presentation. I'd like to invite now Julia Raman Beluca Teixeira. She will be presenting a poster assessment of the activity of the anti-B2A monoclonal. I don't know how you can pronounce these words, swear to God. In Portuguese, it would be difficult. In English, it's almost impossible. Antibody from mexicillin-resistant Staphylococcus aureus against Enterococcus spp. Good morning. She's speaking too far from a mic. We can hardly hear what this lady is saying. Unfortunately, we do apologize in the English booth. We're going to be presenting the monoclonal activity of Staphylococcus resistant to MSR against Enterococcus. What we need to know is that the Enterococcus are bacteria, positive bacteria, and they're part of the microbiota, the human microbiota, and the environment, and also in animals, and they may cause infectious diseases and endocarditis, and these enterococcus present an intrinsic resistance and the BBPs, the BBP5 enterococcus and fecal enterococcus 2, and the staphylococcus are also gram-positive bacteria and human commensals, and they're a part of 30% of the nasal cavity and 20% of the skin in healthy individuals. The staphylococcus presents serious infections and superficial infections. They have sepsis, pneumonia, and endocarditis too. Amoxicillin-resistant staphylococcus have a gene called backyard, and this gene will be responsible for providing intrinsic reaction to the class B of BBPs called BBP2A. Here, just to show you a list disseminated by the publicized by the WHO that talks about resistance to new antibiotics. The aureus and staphylococcus is present in high priority, and this resistance against new antibiotics. About the antimicrobial resistance, it is a global characteristic. When you take the wrong antibiotics, and when you don't treat the infection in the right way, the chances of a new strain cropping up or and disseminating to other organs increases the risk of the global crisis, and also new forms of resistance may spread through people, animals, and other goods as well. Here, this is just to show you a table in which the VRR vancomycin resistance to Heterococcus, the strains of Heterococcus that are most present, and the fecal Enterococcus that are the two main recent Enterococcus resistance to vancomycin. You see that the fecalis enterococcus are more present than the feces enterococcus.
So this shows how the mechanisms shows the reactions. And the PBPs is an enzyme which does the biosynthesis of the wall of the cell, and this avoids from doing the biosynthesis of the cell. And this also, the Staphylococcus aureus has the MAC virus, and the constitutive gene of the allows the talking about resistance of monoclonal therapeutic antibodies. They are important for therapies for cancer, autoimmune disease, and other infectious disease. Our group developed an antibody for monoclonal therapeutic and done by the hybridoma technology, and it's able to work with therapeutic and immunodiagnostics. Our study is that this monoclonal antibody will be able to have a great potential for the immunodiagnostics and immunotherapeutic. Our objectives are to characterize the binding of anti-MRSA BBT2 with the enterococcus. So its first results, we have the expression, purification, and refolding of recombinant protein, where we expressed and we carried out 2 purification processes. So we have expressed and we chose the VES tampon to make the purification. And after the process, we observed that our protein was localized in inclusion bodies. So we were able to purify the first one with a lot of urea, and we had a yield of 0.5 milliliters.
Já na segunda purificação, a gente
on the second purification, we did a releveling of the column where we took the urea before the dilution, and we had a yield of 0.7 per milliliter. As I mentioned before, our monoclonal gene is conserved, so it doesn't have much genic action, but the enterococcus has this mutation presence. So we carried out an assessment to observe the antigen binding with these clones with the enterococcus. So we were able to observe and assess that with these genes could be conserved, and we were able to bind with the clines, the CBP5 presenting polymorphisms. We also carried out a colorimetry assessment showing that the activity of BPB2A was able to bind with PBB5. And as a conclusion, we could obtain recombinant PBP5 as a control. We were also able to demonstrate that the antibody that binds to BBP2A was able to recognize PBP5 even with polymorphism, and that the dissociation coefficient has lower affinity with the monoclonal antibody, with the enterococcus, than with the antibody, the monoclonal antibody. So this is it.
Obrigada, Júlia.
Thank you very much, Julia. You don't know how it is hard to talk. You're doing very well. I don't know if you're aware, but I'm used to microphone and to talking in public. We know you're nervous. And my son, he studies medicine, and he in his presentations he's very nervous, and he has these sweating episodes. And so congratulations to you all. And following up with the program, we invite as a chair of this final Foundation for Translational Research and Partnerships to accelerate the technological development of biological products. Tenure professor of the Federal University of Rio de Janeiro, Leda Castilho. So my son does not study there at your Federal University. She's a doctor in biochemical engineering by the German Research Center for Biotechnology and Technical University in Germany. And she's master, has a degree in chemical engineering by the Federal University of Rio de Janeiro. She's experienced in biopharmaceutical engineering, acting in cloning and protein expression in mammal cells, with the cultivation of animal cells and purification of biomolecules, with an emphasis in the development of advanced technologies for the production of biopharmaceuticals, inputs for tests, diagnostics, and vaccines. And in the Federal University, she works with the chemical engineering at Copy School, where she works with cellular studies. She is also a professor at biochemistry school. And as lecturers in this panel, we call the leader of the division of CNPEM and Sandra Dias. She's going to make the presentation. National Center for Research in Energy and Materials, CNPEM, cutting edge science to tackle global health challenges. She has a PhD in biomolecular physicist in the University of São Carlos, in the Federal University of São Paulo, and she has a master's in biomolecular science in Federal University of Campinas, and she is graduated by the same institution in biology. She concluded her PhD in the veterinary school of the Cornell University, and she works with biomolecular technologies and biochemicals and structural biology to clarify the complex interactions that happen in the progression of tumors. The Vice Director of Innovation of Bio-Manguinhos, Ricardo de Godoi. And with the presentation, Prototype Plant. He's a doctor in science relation in the pathogens and host studies with the Federal University of São Paulo, and also works with the scientific computer, and also works in biology at the Federal University of São Paulo. He works in biogenetics, epidemiological, and digital health and data science, with an emphasis on human genetics, and working especially with population genetics, epidemiologic genetics, and studies of association, binding, and analysis of complex segregation. He worked as a vice leader of the technology of information at Fiocruz, and in the coordination groups in the pandemic of the COVID pandemic at Fiocruz. He was coordinator in Rondônia, the Fiocruz branch, and coordinator in Fiocruz headquarters, and also works in the epidemiological laboratories in the Amazon. And for the online presentation, we invite the Director of Development of Bharat Biotec, Raches Ella, who is going to carry out a presentation for infrastructure to accelerate innovation in vaccines. He's a doctor in the University of Johns Hopkins University, and he has a MBA in Harvard Business School and Harvard Biotec, and he leads the development of products focused on the breakthrough and development of vaccines and therapeutics. So I pass the floor to Leda Castilho. So good morning, you all. Thank you very much. It's very good having this full room for this panel. So as the majority of the audience are Brazilians and the Zoom participants have simultaneous interpretation, I will speak in Portuguese and also to stimulate that you may ask questions and that we have a fruitful conversation here. And I'm going to pass the floor to the panelists, and then we will have a Q&A session at the end, and the production team will send me the questions made through Zoom. So, but I also understand that people can also ask people who are in the room, in the audience. So you can send through the application and also in-person questions. So our table today has as a theme Foundations for Translational Research and Partnerships to Accelerate the Technological Development of Biological Products. And this is a very important theme for Brazil and for the world. The immunobiological is very important for the human health, and it's a moment that is relevant that we are discussing this because the ecosystem that and myself, who works as the Federal University in the cellular cultivation laboratory for the production in biologicals using animal cells. In these years that I have been working with this field in Brazil, I see that we're finally having the innovation sector in this area and the biologicals and gaining momentum and a concrete drive, which leads to the development in Brazil through the whole country, because we have been developing, having developments, but mostly regarding foreign technology. And we see this growth, and these are the consequence of actions that were taken in the past. So in 2007, 2008, under the leadership of the Development Bank and the Ministry of Health, the implementation of public policies and the Development Bank especially identified at that time something that is still real, and we can see some improvements, but it's a deficit in the biologicals and the health field, and they identified that was important to be developed in Brazil, and still currently nowadays it represents half of the expenditures with medication in the country, and we see that we have high value there. And it's important that the public policy resulted during this time in the implementation and deployment of the infrastructure for biologicals. So in this area, we are not only an importer or a packager or a manufacturer for the box and the packaging to distribute. So we have plants here producing biologicals in a greater rate, and then in the synthetics field, which I think that the great majorities are imported, especially from China and India. So in the biological area, these public policies from those years had unfoldings in a way that was very essential, and the partnerships of PDPs for scientific development allowed this development happen in the private sector, but also in the public sector. And Bio-Manguinhos is a great example of this. And we see several partnerships being developed for the unified health system. And this resulted in the creation of infrastructure, expertise, and plants for the manufacturing of biologicals in Brazil. If we can recall, we see that we have this kind of plant in Brazil, allowing that in a emergency sanitary crisis. We saw that what happened in the COVID pandemic. We see that we could have the finished product due to this plant that were already being deployed in Brazil. So otherwise, we would be dependent on imports. So this production in Brazil are still results of transfer technology from foreign companies. And myself, as a researcher in this field, I would like very much to see the development of technologies that are national and produced in the country for us to be able to look at the Brazilian problems, and we could be more cost efficient. And I see this with very much optimism that this ecosystem is gaining momentum and is increasingly important. And I would like to mention 5 examples, and we're going to see 2 of them here. And the third that I would like to mention from these 5 examples, I'm going to very briefly comment on them, but on the Federal University, we started in the beginning of the year to build a pilot plant to produce biologics and recombinant proteins, and to quality assurance, stability studies for pilot batches for phase 1. This is supported by CNPq and with the funding by FAPERJ, and we have the certification. Quality assurance is in a advanced deployment phase, and we imagine that this trivalent vaccine that is being produced is going to be produced in Rio de Janeiro, in the Fundão Island. And this first batch we took our team and equipment for facilities in São Paulo, so we could produce there because our clean rooms were not operational yet. So this is a first example, and we will have a pilot plant in the Federal University of Rio, and also Unesp with Botucatu, and with FAPESP as well. We are installing Cevasp, which is also a pilot plant with CMO and CPO. And they have a company in São Paulo called Bio-Optimize, which are very young people that founded this company, and they established facilities of CRO, and they intend to build a plant to be a CMO. And we have these 2 examples that we're going to see here, which are the LMBIO in Campinas, in the CNPEM campus, and the Bio-Manguinhos with all this structure. We also have this pilot plant, and Ricardo is going to explain to us. And we also have Dr. Ella from Bharat Biotec to talk about his experience in India. So I thank the audience, the speakers, and I pass the floor to Dr. Sandra Dias from LMBIO. Hello, good morning. How are you? Can you hear me? Okay, it's a pleasure being here today. I thank you for the invitation. I would like to start saying that I'm here today very proudly representing our director, Maria Ruda, who we call as Guda. And I'm here on her behalf and the other directors and general director to talk about our institution, the CNPEM, the National Center for Research, and also to talk about the LMBIO and how we believe that we can contribute to this network of research and development in this area.
Realmente é desafio falar olhando para essa tela de ladinho aqui. Eu vou tentar.
It's a big challenge to talk about this subject with such a small screen. I can hardly see what's on that screen. Anyway, I'll be right here. I'll be standing right here if you don't mind. It's a lot better, and then we don't get stuck here. So I don't know if many of you know, but this is our campus, the National Center for Research in Energy and Materials, the Brazilian Center for Research in Energy and Materials. It's a very large campus, 530,000 square meters of surface area adhered to research and technology. We have today 4 national laboratories, about 900 employees, 500 that are not employees, of trainees, postdocs, interns, and other kinds of associated members. What is the mission of CNPEM? It operates through 4 axes, main axes. We have open laboratories for external users. They can, on the internet, we have a page in which anybody who's interested in using those facilities, open facilities, can apply and send projects, and once they're selected, they can come and use the facilities. For academic users, it's free of charge. We have our own research and development that it's done in 5 thematic areas. We develop research in agriculture, health, human health, energy and materials, and environmental issues. We provide support to innovation, our own research, and also research that's done through partnerships with companies. And we have a strong mission in education, training, and outsourcing. This map, we like to use it. It's very provocative. It talks about Sirius, our source of synchrotron. I don't know if you know that, but we have about 80 sources of synchrotron in the world, going from the 4th to the 5th generation. Has to do with the brightness of the source. The Sirius is, together with other 2 laboratories, it's the only, in the case in Latin America, it's the only source of blue synchrotron. But Cirrus and other laboratories are the only 3 laboratories, 4th generation laboratories in the world. We have Max-4 in addition to Cirrus, Max-4 in Sweden, and we have this and Genzel, which is a multi-user European laboratory, in Cirrus here in Brazil. So there are only 3 4th generation laboratories that are the most modern laboratories. Cirrus is one of them. It's a milestone that everybody should be aware of, so we can take pride in it. We have the only laboratory in Latin America. It's one of the 3 most powerful labs in the world. We're finishing up phase 1 implementation of light lines in Cirrus. We're going to have 14 light lines operating. 7 are operational already, and another 7 are being commissioned or installed and being built. Now, on the nanotechnology laboratory, develops research in nanobiotechnology, nanobiomaterials, and devices. I brought you this information because they work with nano and micro manufacturing. A lot of the diagnostic kits that were presented here, I understand that it's obviously in your interest. They can produce by them. They have partnership agreements for that kind of development. We have the biorenewables laboratory. If you want to look into biodiversity and find new enzymes and come up with microbiological plants and deliverables, or to convert biomass into a series of molecules like biofuels and other chemical molecules based on biochemical molecules with the idea of producing biorenewables. And we also have a science school. It's in its third year. The science school has a model that's very unique. All the students selected have full support, full financial support to be able to study at that school. They have housing, lodging, and it works with a teaching model that's not very common in our universities because it's based on the development of projects. And finally, that will be our fifth laboratory, Orion. It's biocontainment, maximum biocontainment laboratory. We're going to be able to work with level 3 and 4 agencies, and that institutions, that laboratory began to be built, and it will be added to CIRUS, and of course, in this laboratory, it will be a large laboratory, large area. It will be possible to develop all kinds of studies related to infectious diseases, and these studies we're going to develop, well, and we say us, we, it's a mission of the CNP and to provide support to research and development in the rest of the country. We expect that Orion and CIRUS, as well as other laboratories, will be open to external users. So we're going to be offering the possibility so that researchers in Brazil and researchers around the world may come and use our facilities for research purposes. The expectation is to develop diagnostic methods, vaccines, discovery of new pharmacological products, and that will be very important for strategic development of strategies, epidemiological strategies. That will be very important for our national public health system, including, and it will boost our sovereignty in terms of new challenges, health challenges like we had one recently. And what is especially unique, what is so unique about Orion that needs to be said? Orion will be the only laboratory at level 4 in the world that will have light lines directly attached or linked to it. What will be these light lines? We'll have 3 light lines for studies of X-ray, MRI, tomography. They'll be called Cibipiruna, Hibiscus, and Timbó. And with these light lines for X-ray tomographies, we'll be able to study cells up to a level of organelles, and we'll be able to study tissues and organs. And we're talking about X-ray tomographies and 3D studies. And we're going to be able to do studies with living animals. It's a unique capacity. No other laboratory at level 4 in the world has that kind of capacity. It's going to be one more reason for us to be proud of it. Why Orion? CIRUS, I don't know if it's the brightest star in the sky, and Orion is a constellation that is close, and astronomically speaking, is close to CIRUS. And in Orion, we have the 3 Marys, with a beautiful poetic analogy between the correlation between CIRUS and Orion, because we have the 3 lines of light going into Orion and providing us with a beautiful analogy with CIRUS, meeting these 3 Marys, if you will. And finally, talking about the Bioscience National Laboratory, what is our mission? To use integrative biology to understand health and illnesses. And by so doing, we hope to provide support to the development of health solutions for Brazil. We're able to work with living matter, from small scale and molecules to organisms. And how can we do that? Because of our expertise in protein biochemicals and biophysics. And that expertise is our core. LMBIO was born as a laboratory providing initial support to our ancient cyclotron source of light. It was a laboratory that provided support to protein crystallography in our ancient source, an old source. That's an activity that we've been undertaking for the longest time. And today we have the ability to work on cells at various levels of complexity, basic cultivations of 2D, 3D, bioimpression, bioprinting, microfilament. We also have a laboratory that works with in vivo models, bioteria, and we have the ability to do genetic additions of these animals. It's an ability that's not very common in Brazil. We have the autonomy to produce these transgenic animals, and of course, computational biology, which today is moving in the direction of using artificial intelligence algorithms to solve problems related to health. We have 5 divisions at LMBIO, 2 divisions that we call Discovery Science. And these divisions of Discovery Science, that's where we do our research. We've been acting, our research groups, a strong focus of our work, research work that we do is to understand molecular mechanisms behind diseases. We're very keen on understanding those disease mechanisms at the cell and molecular level. And they've been working on viruses, bacteria, Chagas disease, and also and there's another division called the Integrated Biology Division, which I lead. We've been doing cancer studies and neurodevelopment studies also, and cardiovascular studies, always using biocomputational research to support this work. We have a division called Advanced Health Technology Clusters, or it's advanced medicine, and in this division we have a pipeline of drug discovery, which uses primarily products from our own biodiversity, natural products for our own biodiversity. We have a park of equipment of mass spectrometry geared to the research. It's very modern and large. We've just installed a new equipment that will be dedicated to single cell research. It will be the first such laboratory able to undertake this kind of experiment. And in this laboratory, proteomics laboratory, the great mission is to deliver and to discover markers and targets for many diseases. And we've begun studies on structural proteomics. We have a tissue engineering laboratory, and it is being moved to another area of tissue engineering. We're going to be able to do 3D cultivation, microfluidity, and we're developing regenerative medicine protocols and genetic research as well. And finally, we're implementing a laboratory of immunobiologicals whose mission is to undertake research and development for immunobiologicals. I'll talk to you now about this laboratory in particular, what we have implemented so far. We are setting it up through the development of a monoclonal antibody. It's a pilot project. We expect that in its full capacity will cater to different projects for immunobiologicals in general terms. Now, excuse me, first of all, I'm going to talk about our 5th division. That's the division of facilities and services, in which we have the coordination of all the laboratories that are specialized at AMBR. These laboratories give us the capacity to do expression, purification of proteins, production of biomolecules such as peptides and nucleotides. We have the ability to do the characterization of these protein molecules as well as others through magnetic resonance, MRI, crystallization, spectrometry, and calorimetry. We have the ability to do high content screening, microscopics, through optic microscopy, and in vivo studies, and cellular culture, metabolomic genomics, and our ability for computational biology as well. Now, talking about our pipeline of immunobiologicals, I will emphasize where we understand we can make a big contribution to the research network in Brazil. It is not always that when you have a target to be aimed at in immunobiologicals, you don't always know how that immunobiological needs to be dealt with, and frequently you need to do a design for that particular immunobiological to ensure it will be most effective. Our mission, of course, has been increasingly We have recently received from our general director the information that we need to go into it strongly with artificial intelligence. We've done that already, but that's decidedly will step on the gas pedal because we understand that these algorithms that have been launched are extremely important. We understand that they won't replace totally, but they have the capacity to speed up research in proteins. And of course, when we're talking immunobiologicals, especially when it comes to proteins, here I just want to bring you information that's food for thought. These data are not ours. They come from David Becker's lab. He is the Pope in the protein design. And this paper came out December last year. And I don't know about you, but I work in structural biology. But now you can design proteins that are not out there in nature around a target. I was impressed by it. This publication came out recently. It's a peptide hormone. They were able to design a ligand, a protein. Again, the protein is not in nature. It was invented by a computer. It has high affinity for that target, with that target, and obviously thinking of the development of immunobiologicals. This peptide can be an antigen, and you can decide you can design better solutions for it. This information that came out yesterday, I had to add it to my presentation. I don't know how familiar you are with these programs, but this AlphaFold came in to revolutionize structural protein molecular biology, and it predicts with high accuracy the spike protein on a coronavirus very accurately. This is a predictive molecule that is very similar to the molecule experimentally seen as the molecule. It brings antibodies, and this is the sky is the limit. And what have we done? We have developed protocols to modulate TCR interaction and antigens. It's been done through cooperation to develop vaccines based on T cell, and we have developed projects as well. Given a certain a known structure of antibodies, the computational pipelines will be able to predict mutations that will improve affinity or modify the way to interact the PCR and the antigen and the antibodies, thereby evolving those antibodies and designing new ones. We have just set up our capacity to add capacity once the sequence has been drawn, and we hope it's the ideal sequence for the therapeutic target. Of course, we're doing genetic engineering to introduce the sequence inside the cells. You need to select, and now I'm saying something that it's basic for everyone. You have to select the cellular clone with the highest production of this immunobiological. We have just installed equipment to be able to do this the selection of these cells, of these clones that have the maximum production of immunobiologicals. We're setting up capacity to expand these colonies, and all of these things that's being done in our own new laboratory. It's beginning to operate, and when it is working in full capacity, we expect to provide research cell banks to the community so that they can develop projects together with us, and in the future that will be done in BPL. Well, finally, this is my last slide. This is the area in which we believe we can make our biggest contribution to the immunobiologicals community, which is the characterization of biological complexes or immunobiologicals and the target. And we can help tremendously on the characterization, especially from the point of view of structural biology, because we have our source of synchrotron, and through this synchrotron source, you can characterize this interaction in atomic level using protein crystallography, and you can do characterization from the point of view of solution behavior using small index of spectrum and secondary, tertiary structures, circular prisms. We have in the National Laboratory of Nanotechnology this microscope called Trotsoscope. It's a very powerful laboratories. We just installed a laboratory, will be 2 such laboratories in the country, and you can do a technique called electronic cryospectrometry to solve the structure of these complexes in atomic level. And finally, at LMBIO, the park of equipment that we have, we hope to have convinced you that we're able to help in various assays of characterization, functional characterization, and the interaction between biology and target using spectrometry and cellular biology. And from the point of view of biophysical techniques, we have plasmonic resonance, light dissemination, fluorimeter centrifugation, ultra-analytical centrifugation, just to mention a few of them. The capacities are many, and again, I would like to I'd like to draw your attention to our spectrometer center, which is very modern, and we can do the sequencing of protein and most traditional research. That's it, basically. I would like to thank you for the opportunity to talk about CNPEM, to talk about LMBIO. I'm leaving you my email of the director Maria Huda if you're so interested in developing a specific project with LMBIO. Get in touch with her to understand how they can operate. Those who want to use our facilities as external users, put it on Google, CNPN, open facilities, and you're going to get to the page and understand how you can apply it to your projects. Thank you so much.
So thank you very much, Sandra. I would like to pass the floor to Dr. Ricardo de Godoi of Bio-Manguinhos, from Bio-Manguinhos. Ricardo, I'm going to let you know when you went through 12 minutes, okay? Okay, good morning, you all. I would like to thank, first of all, the invitation to be here talking to you, to my colleagues. Sandra, we have a very strong interaction with CNPEM, especially with Bio-Manguinhos. We are assembling a strong collaboration. We had the presence of a good part of our team visiting the facility. So I think you have a great opportunity, and it's becoming clear, as Leda mentioned, and thank you very much for your opening, the growth of the ecosystem, and we have to be increasingly closer to our product, of having our products. So I'm going to go through 4 main topics, and this presentation was prepared by Elaine, who is the chief of department of the pilot project in Bio-Manguinhos, but she was not able to be here, so I will make the presentation too. But I would like to thank Elaine for assembling the presentation. So I'm going to go quickly through a vision of what is Fiocruz to understand the context of Bio-Manguinhos, and also a little bit of the pilot plant and the perspectives of collaboration. So Fiocruz is an institution, a centenary institution. We are present in Brazil and several states. We are present also abroad, and we have an intention of acting intentionally, increasingly intentionally, and this presence also reflects in actions in several fields. So we act in the production of biologicals, of pharmaceutical products, and also in teaching and several levels with graduation programs, also that copies the program. We also have technical teachings and programs of communication and health. So we permeate several processes in health. So this very singular aspect of Fiocruz brings some opportunities and challenges for the management of this system. And we have a focus on meet the demands of the Brazilian population and national level, the direct relationship with the Ministry of Health, which we are part of, and we also act in several fields of basic research. So this slide, I'm sorry it's in Portuguese, and I know that part of the audience has interpretation, but I was not able to translate it into English. But we have in the sphere, this system, we have Bio-Manguinhos, which is the organizer of this event, and we are more recent, although the activities that we do there are previous to the launching. So we were founded in 1976, and we had our anniversary last Friday, and we have an expertise in technology transfer, research and development, also clinical trials and other fields. So in this context, these are some of our figures that we have for supplies in the last years. And in this context, we have a very broad portfolio from vaccines and diagnostics, biopharmaceuticals, and we're also crossing other frontiers. And our team is here, Hosanna and others that are worked in advanced therapies and cellular therapies. So it's under expansion and a portfolio that brings also challenges. And in this context, we have acted with responses to challenges and public health emergencies. And we today are dealing with some situations regarding the dengue fever and the situation that we are seeing in Rio Grande do Sul state. So Bio-Manguinhos is always responding to these needs, and I think this is part of the momentum that we have, the drive that we have. So we have to work to have less emergency, and this is one of our concerns, and we think that vaccination is fundamental for everything, for these goals. One of the points of the tables is to talk about partnerships, and one of the partners of Bio-Manguinhos that come from its foundation, acting several products. Then we have a very strong background series, and this is of course summed up, but we have important landmarks that show that these partnerships are being carried out in Brazil and also abroad with countries of several continents. And I think that this movement is essential for the development of our activities. So I'm going to talk about the innovation area in Bio-Manguinhos. We have 4 axes of acting. Of course, there are interfaces and other areas that we work with the providing of technological service, but we also have the research and development with our own products, and we also have the process optimization, which is very relevant for those who have a great number of products in its portfolio. So this is an improvement area that is essential in our context, and it demands a great part of our efforts. And also in the sphere of innovation, the technology transfer field, and also the medical area that we called clinical trials, but have many other attributions from the evaluation of new projects. We have to have this standpoint of the relevance of the products and how they're going to be implemented, and this back-to-end vision sometimes lacks from some of our products. Sometimes we have a great degree of effort for breakthroughs, and sometimes we lack of looking at the possibilities of usage. So, and with the growth of the perspective of great breakthroughs, the access to new technologies is growing, and with that, the challenges of we will talk about in the next table, the valley of death, and they're changing the locus and becoming closer to the end user. So this is a topic that is within intense debate, and which is part of another frontier dimension of discussion that we will have to organize with these new infrastructure and with Brazil, and we talk better within these plants and these partners with technological competence so that we are better aligned and the problems that we will tackle. So in the sphere of, we have several platforms and competence of action, and we are able to act in almost every phase of the technological development, and we are advancing to having a better quality in the spheres of our development, and we are advancing to the best practices in the laboratories and the spaces that we act. So we are working in many fronts, and we are seeking to improve all the initiatives, and their new products are being thought of in this umbrella of initiatives, and this is another new frontier that we are overcoming for the new products. So we get to the prototype plant, and this plant, as I have mentioned, it's coordinated by Elaine, and she has a comprehensive experience in this production area and all the challenges that are connected to this. So we have a virtual tour. She doesn't like very much because it's not it's completely updated, but I think it's good for you to visit and for the communication, and so that people get to know it's a clean environment, controlled environment, and has very restrict access. And we took advantage from our communication unit in situ, and so to open this opportunity so that people could visit virtually. It's not as it is shown, it has evolved from this was made, but I think it's an opportunity to make a virtual tour and to know it from the inside perspective. And we also have a degree of capacity of acting in the level of manufacturing best practices and clinical batches production, and we have been working with the production itself. And we have scale challenges when we get to the biological projects. We are used to deal with vaccines that are billion of doses or dozens of billions of doses, and some of the products that we work are of thousands of doses. And this changes very much the way we do things, and it's very helpful to have this facility to work with this finished product. We have 3 great groups. We work with eukaryotes and prokaryotes and conkaryotes, and a final processing pilot lab where we do the formulation, the formulation, filling, freeze-driving, labeling, packaging, the finishing products. So we work with products that are in the market already, and we work with the new products that we are developing, and we have a possibility of advancing with the new vaccines with mRNA. And the fact that we have this plant, and maybe we will have still 1 year of development and an interaction with a new partner, and we are also able, although it's being developed with the best practices, and we hope it can achieve the best practice in production. So this means new processes and quality assurance. So this experience fitted in this innovation ecosystems is being essential for Bio-Manguinhos, and we hope that with time we are able to adjust and to better use this plant so that we can open it to other partners, and thus be able to expedite not only our own development, but also development of other partners that we have nationally, internationally, and working in this perspective, and we have the installed capacity for the production of new products. And from that was some things that were mentioned today to better talk and discuss with other agents from this ecosystem so that we can accomplish these higher targets and objective to and strengthen the system as a whole. So this was the message that I wanted to pass on to you, and I would like to thank the opportunity to be here. Thank you very much. So thank you very much, Ricardo. We are going to now pass the floor to the presentations.
Ella from Bharat Biotec for the presentation on their structure for vaccine innovation. Thanks, Dr. Ella, for joining us.
Thank you for that introduction. Sorry to switch to English, but I hope you can hear me and see my slides. I'd just like to thank the Fiocruz organization and Bio-Manguinhos for giving me this opportunity to talk to you today about the kind of infrastructure you require for innovating vaccine development. My name is Raches. I serve as the Chief Development Officer here at Bharat Biotech. And before, I thought that I'd frame today's discussion based on a case study. When it comes to innovative vaccines, the most recently manufactured vaccines are the COVID-19 vaccines. And I would like to frame my discussion today in the form of 4 pillars that I think are crucial for innovating vaccines. The first being the vision, what kind of organization you work for. At Bharat Biotech, we are headquartered in India. We are a vaccine manufacturing company established in 1996. We manufacture vaccines and biotherapeutics. And by volumes, we're the 5th largest vaccine manufacturer in the world, where we primarily supply to areas, emerging market areas, that is Asia, Africa, and LATAM. And many of our vaccines are developed in mind with efficacy, safety, equity, and accessibility. Those are paramount important attributes for us towards vaccine development. The second main pillar, nothing can be done in a silo, and collaborations are required. Many of the vaccines that I'll be talking to you today about are innovative vaccines of its own class. And over the last 25 years, since we've been a company, we've established significant relationships with external agencies. These can be academic organizations, government infectious disease research organizations, or also several other companies, as the presentation similar alluded to. When it came to COVID-19, it was a challenging time for everyone, and at that same time, we really didn't know what platform would work, what kind of vaccine would work. Was it mRNA, viral vector, inactivated, or recombinant? At that time, we were not sure. So we started working on 4 different vaccines in various, with collaboration agreements with several organizations highlighted on this slide here. One of the first vaccines we licensed was an inactivated COVID-19 vaccine, which we published significantly in peer-reviewed journals. And we were able to commercialize this vaccine within a year's time, based on the infrastructure we've developed over the years of product development. The third most important pillar is the expertise that the company or the R&D facility needs to have in order to develop such vaccines. The first, under expertise, it's very important to have optionality or the freedom to operate between different platforms. I don't think that one platform fits all kinds of pandemics. I think there is a reasonable case to have multiple attempts at goal. So with that regard, each platform has its own advantages and disadvantages, and it's important for a vaccine manufacturer such as Fiocruz, Butantan, or Bharat to have that availability to pick and choose which platform we would like to use in our vaccine development efforts. With Bharat, we had a significant amount of expertise in viral cell manufacturing. Many of our vaccines are currently licensed with this virocell vaccine for rotavirus, Japanese encephalitis, rabies. We have a phase 3 chikungunya vaccine, a Zika vaccine, which is in phase 2 clinical trials. All of these vaccines highlighted on this slide are inactivated or live attenuated vaccines manufactured with this virocell platform. And it's an extensively studied platform for us. We supplied 300 million doses of vaccines with this platform, and we're really assured of the safety profile of this platform. Another important aspect for a manufacturer or developer to be fully equipped with is its ability to do clinical trials. Usually you need to do clinical trials where disease is actually prevalent. Some of the diseases that we work towards against are not necessarily in India, they're mostly in Africa. Hence, we have to do phase 1, 2, and perhaps phase 3 efficacy trials in the different parts of the world. So it's very important to have that translational ability, not only in your home country, but also outside of your jurisdiction. And since if you're making an innovator vaccine, these vaccines are primarily going to be licensed on efficacy or effectiveness readouts. It's not going to be an immunogenicity or an immune response comparability study. Hence, it's very important for a developer or a manufacturer to have an ability to generate efficacy or effectiveness data wherever that disease is prevalent. At Bharat, we've embarked upon several phase 3 and phase 4 studies. These are largely powered sample size clinical trials in close to 10,000 to 30,000 subjects. And these are all looking at efficacy and effectiveness readouts, be it for chikungunya, cholera, rotavirus, typhoid. We've done a fair share of these kind of clinical trials all across the world. The last main important pillar I would like to talk to you about is the skilled manpower. It's very evident that there are very few vaccine manufacturers in the world. A few noteworthy in Brazil, a few noteworthy in India, and the rest of the world as well. It's primarily because it's a very difficult product to make. If it was that easy, you'll probably have a lot of manufacturers. One of the reasons why we were able to work on an inactivated COVID-19 vaccine is our ability to have a BSL-3 production facility. BSL stands for Biosafety Level 3 production facility, where essentially highly contained viruses can be propagated and grown in these state-of-the-art facilities. As you can see here by pictures of my colleagues during the pandemic, were handling live virus in these labs. And essentially this vaccine that we developed involved growing the virus at large scale, about 10,000 liter bioreactors. That's the scale at which we were growing these viruses in viro cells, and then inactivating them with a chemical agent. Another novel part of vaccine development is the adjuvant development. As you know, adjuvant is a molecule used in a vaccine to trigger an immune response or boost the immune response. And traditionally, there have been only a select few adjuvants that have been licensed commercially. Algel is also known as aluminum hydroxide. It's a very commonly used adjuvant, but it does have a few weaknesses. The first weakness is its inability to generate cell-mediated immune responses. So you really don't have too much of T-cell activity if you use alum. But what we did at Bharat is we tweaked our alum formulation to include in TLR7, TLR8 agonists that led to a balanced immune response. So not only we were able to see neutralizing antibody responses, but we were also able to detect cell-mediated immune responses, which is very important for vaccines to have, to provide short-term and long-term protection. Another important pillar that we had during this process during the pandemic was our ability to collaborate with labs, external labs. These are government labs that could do live viral challenge studies for us. For the inactivated vaccine, Covaxin, I mentioned that we did 2 live viral challenge studies, one in hamsters and the other in non-human primates. Both of these studies gave us the confidence that this vaccine was protecting in animals and could show a beneficial protection in humans as well. So it's a difficult study to do because you have to vaccinate animals to placebo or vaccine, and then you need to challenge them intranasally with live SARS-CoV-2 virus. So select few labs can only carry out these studies. And during the pandemic, it was a very difficult time to actually get these labs available for your vaccine clinical development efforts. And then with this vaccine, we were able to, after the 2 live viral challenge studies, we were able to quickly progress to a phase 1 and a phase 2 clinical trial. Phase 1 and phase 2, as you probably are well aware, you're primarily looking at safety and the immune response in small sample sizes. Where we had to go to larger sample sizes in a phase 3 was to evaluate efficacy of this vaccine, which was reported in a 25,800 sample size clinical trial. We reported a vaccine efficacy of 78% against moderate disease and 93% against severe disease. And this was all done during the Delta variant that affected India terribly during 2020, late 2021. Part of that infrastructure that is so crucial is your clinical trial infrastructure. And the timeliness of doing it, especially in the pandemic, timelines were crunched, not only in product development, but timelines were also crunched in clinical development. And as you can see here, these are some facts from the phase 3 clinical trial that we conducted, where many aspects of a clinical trial, planning of a clinical trial and conducting, which require anywhere between 1 week to 20 weeks, were all crunched down to 1 week to 2 weeks. For example, we conducted a site initiation visit in one site on Monday, and on Tuesday we started enrolling and dosing them. So that was the speed at which we were operating with. And of course, this can't be done only with one vaccine manufacturer. You need to have capable CROs. You need to have capable institutional review boards or ethics committees to ensure that these clinical trials are done at a rapid rate. I'll just like to end by talking about another interesting vaccine that we've recently licensed. It's an intranasal COVID-19 vaccine. And this stems from our interest that the parenteral vaccines, or the vaccines that you inject, fail to prevent infection and transmission. And that was one of the points that stuck with us during the pandemic. All the vaccines that were licensed in the first wave of COVID-19 were all injections. And as you know, they probably failed to prevent against infection and transmission. So we started collaborating with this university at Washington University in St. Louis to develop an intranasal COVID-19 vaccine in the hopes of it being beneficial against infection and transmission. One of the prime reasons why we selected to work on this vaccine is our collaborators working that validated this vaccine in 3 different animal challenge studies. So it's the same live viral challenge study that was done here in mice, hamsters, and macaques. That gave us a reasonable amount of evidence that this vaccine was able to not only protect in the lower respiratory tract, but also protect against infection in the upper respiratory tract. Once we technology transferred this vaccine into Bharat, we were able to scale up the vaccine, and we were able to conduct several phase 1, phase 2, and phase 3 clinical trials, ultimately leading to licensure based on comparable immunogenicity readouts between an injectable vaccine and the intranasal vaccine. We are still trying to evaluate this vaccine, how well this vaccine works against infection and transmission, and we're exploring different kinds of clinical studies. One can also think about human infection models for SARS-CoV-2, which is being evaluated at the moment. To summarize, I think it's important to have, based on the 4 pillars I mentioned on the vision, the expertise, and the collaboration, and outside of product development and vaccine development, where you need to have the resources, infrastructure, and facilities, to manufacture a vaccine candidate, what's gaining more and more importance as we go along this pathway of vaccine development is your ability to do robust preclinical studies, not only preclinical toxicology and potency studies, but also potentially efficacy studies in animals, in preclinical. So these are primate studies, it could be hamster or mice efficacy studies are gaining more and more importance. Clinical trials have become more adaptive and operational seamlessly conducted. One can also start looking at how human infection models, or CHIM models is what we call them. One can also look at how well these studies can be integrated into your clinical development plan. And once the vaccine is licensed, I think that, I mean, there's still work to be done. You still need to be looking at post-marketing safety surveillance studies on these vaccines. And you need to also look at how well these vaccines, at least with COVID, how well these vaccines are working against variants of concern or variants of interest. So it's a broad scope, and I don't think it's only R&D centric. There is a translational side to it that is gaining importance, and there's also a post-commercialization phase that is gaining importance to validate the safety and the efficacy of your products. Thank you. I'll be happy to answer any questions provided this time.
Thanks, Dr. Ella, for the excellent presentation. I'll switch back to Portuguese. Então, nós temos pouquíssimo tempo para perguntas, mas vamos abrir para cerca de 2 a 3 perguntas, idealmente uma para cada palestrante.
Each lecture, ideally. I can see here that online, I still haven't received any questions. Please, go ahead. Good morning, everyone. Leda, I'd like to congratulate you on the excellent work during the pandemic with the development of potential Brazilian vaccines against COVID. Ricardo, Sandra, our colleague from India also. I'd like to congratulate all 3 of you for your brilliant presentations. Sandra, as a researcher, I see a dream coming true at last. And as a citizen, we have to be proud of everything you've done. And now thinking of sandwich scholarships in terms of research at copies, I'd like to know 2 things. The managing board, how does it work to prioritize the projects that get to you? How do you go about selecting projects? And in addition, how can we get our students, for example, thinking initially of CAPS courses, for example, instead of going abroad, it's also important, but they should be developing part of these projects here in Brazil. And I'd like you to talk about those 2 points, if you will, please. Thank you. Sorry, I forgot your name. Joseph. All right, Joseph Paul from ISC and CNPQ. Thank you, Joe. Thank you for the great question. Well, we, as I said before, initially, we have this large mission of offering several laboratories that we have, the facilities to the community of users in Brazil, academic users and corporate users as well. And the way these projects are selected ends up being a bit within the format of each laboratory. Each laboratory operates in a certain way, and it's a distinct manner. And for some facilities, basically, you apply for a project, and you have to do a selection, and that has to do with the complexity of the facility, the number of proposals that are sent. So for the synchrotron laboratory, there are committees geared to assessment made up of internal researchers as well as external users, and these committees are specific to each line of research. Projects are sent, they're assessed, and as a whole, and it's a comparative analysis are selected, and we do the public publicizing of these projects. At AMBR, we have laboratories that are open to users. We want to expand them briefly. We will have another 4 laboratories or 6 in a total of 10 laboratories. And again, each laboratory will have its own dynamic depending on the flow of projects that will be sent to them. How then do students in Brazil and the universities in Brazil, how can they access our capacities and facilities and develop their projects? One of them is to apply for projects, and then this model, a student, that happens. It's been happening for several years. CNPEM operates for over 20 years, at least 25 years, I would say, and since our first look from source of light, we've been catering to students from many institutions in Brazil and abroad throughout these years, and to operate projects in a more complete way. We also have models where, through partnerships and cooperations, students come to our institution and they can spend months undertaking their research projects, and that ends up being at the discretion of different research groups which work together. It depends on the context that are established. In LMBIO, under the new leadership, the expectation is to issue calls for projects in the sense that these projects will be carried out almost entirely at the laboratory. That's what we hope to have in the future. Thank you, Sandra. We have the time for one more question. Congratulations for the great panel discussion. My name is Renato Estrella. I'm from the Butantan Institute, researcher. Ricardo, I'd like to know, in your pilot plant, the pilot plant that you have, we're thinking of a business model now. I think that you're planning to have, do you plan to have partnerships or services? And how can you compete with these services, external services hired abroad, vis-a-vis costs and speed? Because we know that the speed here for us to acquire inputs for new projects, that could be a hindrance to us. It could be an obstacle. How do you overcome that obstacle? Have you had it mapped out? I'd like to draw on this opportunity to say that Paulo Lebo's right there at this point, at this moment. That's why he couldn't make it today. Deal with a partnership that we're trying to establish and exchange information on the production of RNA. We're developing this model literally now with you, and what we first of all we're trying to stabilize the internal use. We are a relatively new facility, a young facility, and starting through a strategic interaction to receive our science and technology projects. We're launching this model with Butantan literally today. Paul is there. He has Eleni Patricia with him, the coordinator of the project, and we will be seeking out a qualitative differential. We've done improved process improvements and the development and the manufacturing of our prototypes of mRNA, and that's how we're going to be trying to gain ascendancy and building a technological differential that's innovative in the production process. We have the first facility, GNP, it's in its final phase of engineering implementation, a new platform that will do the automated production, and that's how we're going to seek out that new route. We're not trying to, first of all, we're not seeking competition in terms of volume or financial sums. That's how we're going to develop the relationship with Butantan. As of today, we're going to sort out the main issues, but the initial focus will be on strategic projects for the country and the organization as a whole. I think we're materializing this project today. Hopefully next year, we'll be able to tell you about this whole story in more details. Thank you for the invitation. Renato, perhaps I could contribute to what you just said. During the pandemic, we look for budgets from many CROs abroad for different parts of the development of vaccines. And we know that the costs are staggering, millions of dollars. And that's stuff that we ended up doing in Brazil for a small fraction of those costs. So that there's huge potential. And also we have shared with Elena Caridi and other researchers, and we're able to find the certification of cellular banks, and we obtained bio-reliance certificates, and we found a network in Brazil with several laboratories that could do, at least in a phase 1 of clinical studies, at very low costs, and there was a line abroad. So it's a matter of time. We were able to do it at much lower costs and reduced time frame. We were able to do things that are acceptable from a point of view of the meetings we had with Anvisa and so on. And I think that that's the question of following what Ricardo said, trying to come up with a network among these institutions, bringing them all together. Pharmaceutical industry does a great deal of outsourcing, even large biotechs in the world, large pharmaceuticals in the world, they do outsourcing of various R&D stages and development and research. RNBio presented what we can do for Bio-Manguinhos as well. I said, I talked about the many institutions that are being commented upon.
Biotec, os NB3 para fazer estudos de desafio, para ver eficácia de vacinas. A UFRJ tem um NB3 multiusuário. O segundo criomicroscópio que tem no Brasil, que você falou também, é multiusuário na UFRJ. Então, para além do nosso laboratório, da nossa planta piloto, a parte de caracterização, de ensaios de desafio também, a UFRJ está disponível. E eu acho que a gente tem que, então, seguir. Vou finalizar com essa mensagem que, na verdade, veio de um cochicho ali do Ricardo, né, que a gente tem que fazer uma rede dessas instituições que podem, a nível de Brasil, trocar experiências e encontrar sinergias para que a gente consiga desenvolver cada vez mais a área de biológicos no Brasil, porque desenvolver a área de biológicos no Brasil significa reduzir custos para o SUS e aumentar o acesso da população a esses modernos medicamentos e vacinas da biotecnologia para a saúde humana. E acesso da população à saúde, né, é o que tem que ser o que nos move. Né? Então, queria agradecer muito a participação de todos, né, e finalizamos assim nossa mesa. Obrigada aos palestrantes.
Obrigada, Leda, pela sua moderação. Obrigada também aos palestrantes. Pausa para o coffee break, mas não deixem também de assistir aos posters e visitar os estandes. Tá bom? Voltamos em 30 minutos. Até já. Acomodar, por gentileza.
Hello, please take your seats.
Bem, dando continuidade, então,
Well, moving forward with our program, we invite as chair of the panel, the Vice Director of Bio-Manguinhos for Innovation, Ricardo de Godoi. For the presentation, presentation, we invite the senior scientist for the bioengineering for Max-Planck-Institute for complex systems, Yvonne Genzel, who will give the presentation Next Generation Virus Production from Clone to AMBR to Perfusion Very High Virus Yield. She's a doctor in biocatalysis by the Mediterranean University in Marseille, France, and also postdoctorate in Von Goethe University in Germany, where she acts as a professor. And in the Max Institute, she leads the upstream processing team with the production with mammal cells. The Vice President Associate of Strategy for Investment and Development of Portfolio and Innovation of Global Health, Innovate Technology, Hayato Urabi, who will make the presentation Vaccines for a Safer World, the Pitfalls in the Development of Vaccines for Neglected Diseases. He concluded his bachelor in molecular biology, has a master of entrepreneurship engineering, has a doctorate in biomedical engineer from Brown University. He has an additional master's degree with global policies in the University of California, San Diego. And he acted in finance and technology areas and strategic planning and due diligence of startups and project management in the Silicon Valley. He was a consultant for Syntech Genomics for technology that goes from neutral genomic until biofuels. He oversees all the activities of investments and access, such as exploratory activities to seek for technological seeds to apply to global health and its financial models that are innovators. We invite to the stage the professor in technology innovation projects from Getúlio Vargas Foundation, Paulo Negreiros Figueiredo. He will make the presentation, Managing Innovation in Human Health Biotechnology Startups in Brazil, Evidence and Lesson Learned. He's a doctor in Science, Technology, and Innovation by Success University in the UK. He's a senior researcher at Oxford University and a founder and responsible for the research program in technology and innovation of Getúlio Vargas Foundation. So I pass the floor to Ricardo Godoi. So good morning. I'm here again with you. I was here in the previous session, and I would like to thank the presence of the guests here. I had the opportunity to talk a little bit with Yvonne and Ayato online, and I would like to thank we have a speaker from SGV in this space that is being very good for a symposium. This is a subject that has gained the visibility and attention and regarding innovation for to the government for the efficacy that is expected, and we have given a very fair amount of attention for this subject, and also we have been working with implemental science. And I would like to confirm with the speakers if the translation is okay, if you are receiving the translation. Okay, are you able to understand? Because we speak English, but I know you have interpretation, so I can see that you can understand me. The translation is okay, right? Can you confirm, please? I think that Yvonne may be...
Tudo bom.
Tudo bom. So thank you for joining. Good evening for you. I think it's good afternoon for Yvonne. And it's a pleasure.
We'll just pass the word to Yvonne to start. Yvonne, please, if you can jump in. And I will take a close look on the time so we can have more time to discussion with the audience here. Thanks.
So thank you. I hope you can see my screen. I have no view on what it does right now. Can you see the screen, Ricardo?
Yeah, we can see the screen.
Okay, thank you. So I would like to start by thanking the organizers for giving me the opportunity here today to give you my idea of how I see that we can overcome the valley of death. I will start, by explaining that, well, how this goes for virus and vaccine production. So at the beginning, you have a virologist or a medical person who has an idea for a new vaccine. He's producing this in a small scale, typically with adherent cells in a tea flask, and he's happy to have 1 × 10⁶ infectious variants per milliliter. Then he tries first experiments in mice. For that, he needs to purify the material, and this is typically easily done in centrifugation. And then comes the next step, and this is what everybody always refers to, the valley of death. You have the idea, and now make it to a product. So what you will need an easy, scalable GMP compliant process, and you are targeting to much higher virus concentrations. So for getting into vaccine doses of 1 × 10⁷ or 10⁸ variants per dose, or for gene therapy and virotherapy even higher, you are aiming for producing in your bioreactor 1 × 10⁻10 infectious doses typically. For that, you will need a high producer cell line, and doing this with your adherent cell line that you have in your lab will probably not be okay, because you need a qualified GMP-ready cell line, and if possible, it should be a suspension cell line. So the technologies today are emerging, and we have already heard it in the previous presentations this morning on what great laboratories that are there at the CNPEM to be there. So I would like to give you a flavor that actually I see it not as a valley of death, but a sea of opportunities that we have today with all the technologies that are available. So if you think of producing a vaccine with adherent cells, you grow the cells typically to 2 million cells per ml, and then you infect them with virus. If you do this with what we heard this morning with adherent viro cells and on microcarriers, there you do typically also medium exchange. And yeah, if you do this with suspension cells, your starting material comes from shake flask, and here, this is already good. You can today grow these cells up to 10 million cells per ml. With the new packed bed bioreactors available, you can, with adherent cells, easily also reach higher cell concentrations than that with microcarriers. Disposable bioreactors available today are 4,000 liter ones, and bioreactors up to 2,500 liter scale. but also microbioreactors are there. And this can ease up your process development. So these are small bioreactors of the size of 15 milliliter working volumes, and you can run 48 bioreactors in parallel. On top of that, there is now also automated equipment to do clonal selection. This is not so much done in virus production yet, but for monoclonal antibodies, cell line development, this is used a lot. And I would like to demonstrate to you on what this can do. And finally, what I want to show you here as well is that you can today also grow suspension cells with the respective cell retention devices easily up to more than 100 million cells per ml. Why is this important? If we look at the viruses that can be produced, so the ones that we work in our lab mainly, for example, influenza, modified vaccinia ankara, yellow fever or Zika, also a vesicular stomatitis virus, you see they all have different shapes, different sizes, they have a different genome, they replicate differently. But moreover, if you look at the cell-specific virus yield, so how much the virus produces per cell, you can already see that there's also quite a difference. So in respect of infectious particles, for example, yellow fever, if you are happy, you get 20 variants out of one cell line, of one cell. So, and at the end, you need to match the cell line you're using with the virus that you want to produce, and not every cell is producing every virus. So we are lucky with a long collaboration record with many, many partners. We have been now collecting several suspension cell lines in our group that you see here listed. And if you now take such a microbioreactor and you do parallel experiments, you can do a quick screening of all the cell lines you have available. And this is what you see here. So we have tested for a yellow fever construct all the cell lines we had, and the reference in adherent vero cells is the red line here. You see that already by screening without optimized condition, we were doing better than the reference. We continued by taking one high producer, one middle producer, and one low producer cell line, and tried to add viral sensitizers. These are small compounds that you can add at time of infection, and they have an impact on the virus yield. But you need to screen them. So we did a screening of 8 different compounds together with the company Verica. And here you can see that we were able to increase the yield even more. So first of all, 100-fold increase by just screening, and then another more than 3-fold higher. I said I want to show you about the clonal selection. So this was a cooperation with Sartorius where they did the clone generation, and they gave us 10 different clones to compare. You see here what the machine is doing. So you really start from one clone, and then you expand them, and then you can test it. And we had the task to test these cells for influenza virus production. We weren't sure if this was really possible to be able to see which one was best. So we first performed shake flask experiments just to see on the growth. So some of the clones were just reaching maximal cell concentrations below 5 million cells per ml, but some were really outperforming, reaching up to 15 million cells per ml. So really high growth. When we then tested in the AMBR microbioreactor again, for virus production, here you see the infectious virus titer and the HA virus titer, which is the important one for influenza. For total virus yield, you could see that the clones were doing quite different. And if you try to put this into numbers and compare this, so here we look again at the cell-specific virus yield here in total virus particles, you see that one clone was really outperforming all the other clones. So it makes sense to do something like that to already give you a good starting material. Now trying to grow into high cell concentrations. Today there are several retention devices helping if you are growing cells in suspension to retain them in the bioreactor or to remove the medium and to give the cells fresh nutrients and fresh medium. Some of them are membrane-based and some of them are non-membrane-based. All you have the retention device attached to the bioreactor in the outside. Here you can see how this looks like, connected to an orbital shaken bioreactor in our lab. This is a hollow fiber membrane that is doing the cell retention. And by using this membrane, we were able to grow avian cells from the company Probiogen up to more than 30 million cells per ml. In comparison, without the cell retention device, you see here the batch cultivation just of 1 million cells per ml. So if you now look at the cell-specific virus yield, it is important that you're able to produce the same amount of cells per viruses per cell as you can do without the cell retention device. And we could even show that we were outperforming and getting better cell-specific virus yields. And you can see that we were reaching very high yields, up to 1 × 10⁻11 total virus particles. We also tried this for another virus. This is for yellow fever and for Zika virus. This is also an avian cell line that we received from the company Valneva. And here we were also trying different perfusion strategies, so feeding strategies during this cell retention. And with our best run, we were able to grow these cells up to 160 million cells per ml. And this is the typical titer value that you get normally. And here you see that we were obtaining 1 × 10⁻10 infectious virus particles per milliliter. And again, we were able to increase the cell-specific yield. So we think that with this system, out of 1 liter of harvest, we were able to produce 10 million yellow fever doses. Finally, I want to present to you a work that we have done recently on a vesicular stomatitis virus. This is a virus that fuses the cells before they lyse and before the virus gets out of the cells. So you get these big particles in your bioreactor, meaning that running this with a cell retention device, membrane-based, could give you some problems. This is in collaboration with a group in Munich, and they came to us and said, yes, we have the virus growing to 1 × 10⁶ in adherent cells. Can you try to bring it to 1 × 10⁻10? We did a first screening, and in this case, the BHK21 cells were the ones that were performing best, and we optimized the whole process for this into a cell retention perfusion run, and finally used a new membrane, which is a tangential flow death filtration membrane, and were able to perform here also grow the cells to 20 million cells per ml, and then finally almost produce 1 × 10⁻10 virions per milliliter. So with over the years, with a lot of collaborations, we have been able to grow different cells that are good for virus production to very high cell concentrations. We have cultivated them in different bioreactor setups with different cell retention devices, and we tried different viruses. And as you can see from here, we are able to obtain very high virus titers by that. And so this is how we think that next generation virus production and vaccine production should go for. And I hope I have convinced you that all these new technologies really are giving you a new sea of opportunities. And what I heard this morning in the Innovation Hub, I think you are very well prepared in Brazil to take this route on and to make this happen. By that, I want to stop with my presentation and acknowledge all the cooperation partners and my team in Magdeburg. Thank you.
So thanks a lot, Yvonne. The session is structured in a way that we will go for like a technological improvement perspective. Then we will start to discuss a little bit more management and business strategies. So that's why we do have different topics. And it was great to have your presentation, Yvonne, that deals with the very first steps. We need to have good technology to move forward, and we will need to have more and more better systems to move forward. So now I would ask you, Ayato, to jump in for a presentation, please. And Yvonne, please stay with us.
Great. Can you hear me? Can you see the slides?
Perfectly, yes.
Yes. Great. So I will start. Bom dia, everybody. Obrigado, organizadores, por me convidarem para a conferência. So this is my. So thank you very much. I hope I'll be there in person in one of these coming years. So I'll be talking about vaccines for safer world and pitfalls for development for vaccines for NTDs from a funder's perspective. So and I'll be giving a little bit about the introduction of what we do as a Global Health Innovative Technology Fund, or GHIT Fund, which I manage the entire portfolio and investment process. So we and then I'll be giving a little bit of the one of the investments that we have recently made in terms of these vaccines in the NTD space. So GHIT Fund, we do have a vision of a world free of neglected infectious diseases that affect billions of people. And we are trying to leverage the Japanese innovation in collaboration with innovations outside to solve neglected infectious diseases and free that from the burden from the people who are suffering from them. In fact, what we do is we do make investment in R&D, and so that the product development would have this cycle which would stop the onset of different diseases and increase the productivity of people in those nations who are suffering from these. Just to start off with where we're coming from our funder, we are quite unique in a sense that we're getting funding from both the public and private sector. From the public sector, 50% coming from the Japanese government, and 25% were funded by the Gates Foundation and the Wellcome Trust. So the foundation's providing us 25%, and then the rest 25% coming from private sector. And this particular model of public sector and private sector matching the fund is actually being replicated in places like Korea. There's a Wright Foundation, which models exactly the same. And also you're probably familiar with CEPI, Coalition for Epidemics Preparedness and Innovations, which also working with this model public and private partnerships. So we have invested over the last 10 years over $300 million for product development in the area of malaria, TB, and neglected tropical diseases, which I will talk a little bit more on. But as you can see, our interventions vary from drugs, vaccines, and diagnostics. And of course, vaccines is our main topic of discussion today, which covers about a quarter of our portfolio. And we do cover everything from discovery to clinical end. And we have partnered with over close to 200 partners around the world. And you can see how we have some presence in Brazil as well, with work with Bio-Manguinhos in one of the drug projects that we have. I'll skip the slides for now very quickly, but just not only do we do R&D, but we do actually connect with access and delivery, which is going to come to this the thing about this valley of death, which our previous speaker has spoke a little bit about. Of course, NTDs in vaccine space, we do work. We have funded multiple projects, and in the space of Chagas disease, dengue, leishmaniasis, and leprosy. These are the NTD vaccines that we have funded up until now, ranging from preclinical or lead optimization stages up to clinical phase 1 or 2A for the case of leprosy, and we are actually seeing a lot more promising projects coming through. And just referring back to our previous speaker, Yvonne, with respect to the Valley of Death, I think there are multiple valleys of death. I think it was a very good start that we started this session with the translation R&D gap, which was very well elaborated by Yvonne. But I think there's also other types of valleys of death with regards to approval or policy of introduction challenges. Of course, vaccines, they have to be injected into healthy individuals, and it does carry that additional usability burden, etc., for introducing these interventions on the ground, and also the valley or the line that you see at the bottom, the insufficient funding. Unless you have the funding for doing these interventions, there's always that you cannot advance some of the R&Ds that you would have. So there's that inherent valley that's at the that underlines all the factors, as well as the third valley being the access and delivery challenges. Even if you have the product that's already registered, how can you actually bring it to the ground? And that's another valley that I will touch upon a little bit. Just going through the first valley of death, translation R&D gap. Of course, I'm not going to belabor the point that Yvonne has mentioned with regards to the complexity that's actually there, coming from translating from R&D space to actually moving into product development stages. Of course, it's very important for us to prioritize an early vaccine development pipeline, identify the gaps, and align the target product profiles, which we really do emphasize for that investment that we make, to know where you're exactly targeting. And if you don't know where you're targeting, obviously you're not going to be able to make the product where you're aiming for at the end. So not only just start with R&D, but have the end goal in mind is a key to jumping over this valley of death. And with regards to second valley of death, or approval of the policy of introduction challenges, of course, there are multiple regulatory agencies around the world, and each regulatory agencies around the world, not only the FDA or EMA, but also the WHO listed regulatory authorities in how you're actually going to be approving, especially for NTDs, if they're endemic. If they are in endemic countries in different regions, you have to know about their regulatory cultures or regulatory procedures in order for you to navigate that space to successfully enter some of these products going in. With regards to funding, of course, we're only part of the puzzle. We do make funding for NTDs in vaccines, but someone like us just being able to fund product development, but also looking at some innovative financing mechanisms like such as the PRVs or priority review vouchers, or knowing advanced commitments of the vaccine purchases by different entities is also another way of looking at ways to tackle these insufficiencies of funding. And of course, access and delivery challenges is a whole talk in itself, but you have to be able to understand, even after you have the regulatory approvals, how you're going to be moving from that technology that you have that's been regulatory approved, but then bringing into the different countries, because you do have not only if you have to consider the regulatory approvals, but you have to think about the usability and how they're going to be procured, stockpiled, and delivered onto the ground. So there are multiple things to consider ahead of time. Now, let me go back to a little bit of the sciences in terms of what we have invested in this leishmaniasis vaccine. In the world, currently, there are 3 major pipeline of vaccines for leishmaniasis, of which you can see, 3 of which you can see here. The one that I'm going to talk a little bit on is the what you see in the middle, the centrine deficient leishmanin vaccines, the live attenuated vaccine by Genova Biopharmaceutical in Japan in collaboration with Nagasaki University in Japan. Of course, you know very well about leishmaniasis and its impact. You can see the geographic expansion of how these are shown in the world. And this is, if you have a vaccine tool, even with the 50% blocking vaccines, it's modeled that we'll be able to substantially decrease the amount of the VL that's endemic in the world. Even with the 50% blocking, you can actually go down with that level of elimination. And this most effective vaccination strategy so far, at least this is what our grantees claim, is it is possible by having these live attenuated vaccines, live attenuated leishmanin, where they would have the centrine gene taken out using CRISPR technology, where they would be able to achieve this genetically modified attenuated parasites, which is not going to cause infection, but you'll be able to have protection. So it's an old technology brought to life again, but using some of the gene editing technologies that are more cutting edge that weren't available back in the days, so to speak. It is a genetically modified attenuated vaccines. It's on the verge of getting into phase 1, but they do have advantages over the other approaches. And let me show you a little bit about some of the initial animal data, so to speak. So centrin gene, you're just removing centrin gene. For those of you who are participants in this panel, you know about the CRISPR-Cas9 technology. You're basically editing the gene out, and then you're doing the in vitro growth for this wild strain and the centrin deficient gene, and then you're making sure that with the whole genome sequencing, you make sure that the centrin gene is taken out, and you are immunizing these centrin deficient leishmanian parasites onto the animals, and then you're trying out with this sandflies. Basically, you're infecting them using this particular vector. And what you see here, this animal model product, what you see on the top left, the immunized mouse versus a non-immunized challenged mouse. You can see the weight loss being seen for the hamsters for visceral leishmaniasis. And then, of course, you can also see the cutaneous leishmaniasis in mice by looking at some of the lesions that are being affected, with the comparison being shown on the bottom right in the graph there. So just as a summary of this particular study that was done by Professor Saroskar in Ohio State University in collaboration with Nagasaki University and Genova, who is basically doing the manufacturing of this centrin deficient parasite, is that it is safe, and it has been shown the immunogenicity with the high efficacy with, as I said, it's the second generation of leishmanization, bringing up to speed, basically combining old technology, but combining with the new gene editing technology onto NTD space to create this particular parasite. Now it's planning to be tested in phase 1 in the coming months. So just concluding, I kind of rushed through this, but just coming back to this, of course, we talked about the valley of death, the translational science valley of death, as well as there's a second valley of death about the regulatory valley of death, which requires alignment of the regulatory policy financing continuum. But there is that portion of the financing valley of death where that sustainable vaccine development will be key by using some sort of a collaboration. And of course, we, as ourselves, GHIT Fund, we're just one piece of a puzzle, which we would welcome any of the participant who is in this room to contact me if there's any interest in our work, as well as any potential synergies that we obviously observe for some of the programs that we see. Because we do make investments in not only vaccines, but also in diagnostics and therapeutics, we have seen some programs being able to be bunched up together to tackle the program together. For instance, the leishmaniasis vaccine that I mentioned at the end, that particular technology is being applied for leishmanian skin test as a way of testing whether the patient has been subjected to has had leishmaniasis before, being able to use that as a diagnostic as well. So we're trying to look for innovative ways to combine our technologies together. This last slide here, just, I don't know, maybe it's just a self-promotion in a sense. If you're interested in finding out a little bit more about us, please go to this QR code. If you sign up on the newsletter, we'll be able to send out the next request for proposal, which is going to come out in June this year. So with that, I'll acknowledge Professor Abhay Saroskar for his work on some slides, as well as my colleague Cecilia, who has helped me create these slides. So with that, I'll pass over back to the chair, and muito obrigado.
Muito obrigado, Ayato. Vejo que o seu português está bem avançado.
Has improved remarkably. And thanks for your presentation. I'm going to give the floor now to Paula Negreiros. You can make your presentation from the pulpit, from the lectern over there, and we'll move on to Q&A right afterwards. Good morning, everyone. It's a great pleasure to be here with you. I'd like to thank you at the Getúlio Vargas Foundation and Fiocruz for the chance to participate in this symposium. We have a research problem in technological learning and industrial knowledge. I've been a leader at the Getúlio Vargas Foundation involving the management of the process of innovation. It's a set of startups in Brazil in the area of biotechnology for human health. I've had the privilege to lead that study with a group of researchers that are highly talented, involving bringing together, for example, bringing together Professor Daniel Oziel from the Federal University of Rio de Janeiro, Marcela Cohen from the Federal University of Fluminense, Pedro Gomez, a master's student at the State University of Campinas, and the assistant researcher at the Getúlio Vargas Foundation, Lohana Palini, postdoc at the Oswald Cruz Foundation, and Bernardo Cabral, a professor of economics and biotechnology at the Federal University of Bahia. That is, we have an extremely varied and distinguished group with contributions that are very important to this study. The motivations for this study mostly involved facts that even in the advanced economies, investments, high-risk investments have not been geared to the science and health studies. They've been geared to digital technologies and thereby avoiding risks in the health arena. Even accelerators have, in a way, they haven't felt so attracted or drawn to the area of health sciences. Hence, many ventures and startup initiatives have not gone too far due to lack of investments. And certainly also in the literature on innovation, there is a scarcity of studies involving the process of innovation and biotech startups and how they may evolve till the stage of commercialization, successful commercialization. This scenario has been aggravated, has been worsened in the context of developing economies, which is the case of Brazil, in which we have over 200 million, and we have a demand, an increasing demand for public health services. And we have, because of the universalization of the public health system in Brazil, there is an increasing demand for therapies and biotechnological therapies and medications, and certainly for pharmaceutical inputs. At the same time, the country has a very low supply of pharmaceutical inputs and biotechnological products, which leads to high imports, large volumes of imports, and that has been even greater at the time of COVID, a greater dependence of imports, leading to a commercial trade deficit of over 7 billion reais in Brazil. And even though we're the sixth largest pharmaceutical market, we only produce 5% of its inputs. We produced 80% of them in the past, but we have fallen down in that production. There is an urgent need to better understand the ecosystem of biotechnology for human health, to try to see how we can strengthen it. That's why the objective of this study is to understand this process of accumulation of capabilities to produce and innovate in biotechnology for human health in the Brazilian ecosystem for health and biotechnology, especially at the level of startups. The analytical model of the study basically involves three large variants. The central variable basically is the level of capacity, technological capability, which is a set of resources that will allow companies, startup companies, and organizations and institutions to undertake production, and also innovation. That's a central variable, and we would like to understand the sources for these capabilities and the results, the outcomes, the impacts that the accumulation or lack thereof of these capabilities have generated. Basically, our study has involved a set of 18 startup companies in four large areas: and in vitro diagnostics, startup molecules, gene therapies, and tissue therapies. And they have a time, they've been in operation since 2008. Some of them are younger, 2021, for example. The names have been codified because the startup companies, their names would not be publicized. We have an agreement with them. We have a very interesting set of startups, even though the sample is not too large, but the study has been in depth and they have offered rich evidence, wealthy evidence to illuminate public policies, to shed light on public policies. They are scattered around the southeast and south region of Brazil, especially in the state of Sao Paulo. And the study started in 2021 and involved a series of steps going through recursive processes, coming and going, and so on and so forth.
So that we could obtain evidence, in-depth evidence, and qualitative depth also is this. And the academic studies that review literature, they obtain from the companies the information, and the final, they generate a paper that will be delivered to society. So we seek to involve these startups since the beginning of the study so that they could help us with the design and the questions of the research. So we made several workshops with them to feel the problems and the main issues and their pains, and that this could be embodied in the design of the study. So they followed through all the study, which gave us better connection with this important side of the demand. In terms of business model, the startups are distributed among several different business model, also the hybrid ones, which involve product platform and products, and their investment are basically that come from governmental bodies such as the Development Bank. And if we opened that part of the investments, we will find those that come from the state agencies for innovations, such as Embrapi and FINEP. And we also have multi-strapping investments. So going to the main variable, which is regarding technological capability, we understand this is a storage of knowledge that the institutions accumulate to generate innovative action. So there's a set of knowledge that distributes, such as human resources skills, business skills, professionals, and technical and physical systems, and so on. In the case of the startups, we prevails the human resource in the researchers and the professionals, and the others are more well distributed in the ecosystems. And the idea when we examine the technological capability is not if they have or not, but the degree in which it exists. So this is why we measure the level of technological capability in these startups. So we avoid the conventional proxies in literature, and we identify this maturity model that is based mainly in the revealed capabilities. That is the capability of the startups revealing their activities and what they're able to do. Another distinction is where you differentiate the production and innovation. One thing is producing of vaccines with an existing technology and you're user of this technology, and the other thing is to develop the capability to adapt this vaccine and even more scale up and create a new vaccine. So we identify these levels and up to which point these startups develop themselves in the capabilities. These are the results, and from these startups 2 and 4 reach the levels 2 and 3, so they are able to adapt in existing technologies, and with a new degree that is for themselves or in the regional context, but 7 of them advanced to the advanced level next to the international frontier. But here we have, gentlemen and ladies, 5 startups that could reach world-class level, and 2 of them went to the commercialization phase. It's a great merit considering the concept that we have, the hardships that they face to operate in a country like Brazil to reach this level, carrying out activities that are new to the world in a very difficult area. So we have much to learn with these startups. They generate interesting evidence to public policy generation and how we can support these technological capabilities and not allow to these levels weaken themselves and get lost in the ecosystem. So we should also look at these 5 that could reach this level 5, and some of them are hybrids, and the type of investments that they receive are mainly for FINEP, CNPq, and FAPs. And in terms of the partnerships that they establish, and they carry out many partnerships that are local, universities that are local in Brazil, also with specialized consultants and private international research centers. So the localization of these partners are Latin America, followed by North America, and this axis is from where they seek the knowledge to they can carry out these capabilities. They use external sources for such as training abroad, partnerships. Some of them are based in research and development, some are not, but they also carry out internal efforts for knowledge creation. So going to the results and the outcomes and the levels of these 5 startups, we have outcomes in terms of patenting activities, some in the US and also in Brazil, and also results in terms of technological activities in the production and markets. So in the end of the presentation, we're going to compare those that were able to achieve this commercialization stage against those that were not able to do so. So in terms of partnerships, they all can make partnerships with specialized consultants and also with local universities, but there's a difference. Those who could get to the commercialization phase, they have very important partnerships with pharmaceuticals and biotech institutions, in addition to having these partnerships with the local universities and specialized partner consultants. So there are clues that you can observe to get to the commercialization phase. So the regions are Latin America and North America, and the efforts that they make are basically of international PhD, international collaboration, but also local and also internal efforts of knowledge generation. So we can see a distinction in terms of efforts of learning that for those that could reach their commercialization against those that could not reach this level. So regarding results obtained in this commercialization stage for respectable patents in the US and Brazil, and they could also reach market outcomes for these that have reached this level. So these studies allowed us to issue some recommendations in terms of ensuring the needed funding for them to generate the innovative activities, the investment in practicals, in learning practice to get to this level of commercialization, get to higher levels of technological capabilities. Also, these develop partnerships with biotechs and pharmaceuticals is also very relevant in the process, and the development of capabilities beyond technical and scientific. So technical and scientific is essential, but it's not enough. So there you need to develop the skill, management skills, marketing, entrepreneurship skills. So adjusting the business in itself, that's why it's important, this partnership with the management training. For public policies, we strongly recommend the simplification of access to funding, which was an issue that was very much discussed by the startups, the difficulty of finding pathways, of the lack of integration among the organization that operate the fundings, and also consider the capacity of the innovation of a main activity in this process, also to support startups to go through the go up to the commercialization phase, and also to expand this commercial skills. So I'm here. Thank you very much. Thank you. I'm available to answer your questions. Thank you very much.
Muito obrigado. Acho que é excelente, né?
Paulo, thank you very much. The study that you carried out is excellent, and the previous organization who seek these spectrum of different initiatives, and we're having a tight time here. We should end at noon, but I will ask a little bit of patience. So we have 3 minutes to have the opportunity to have 1 or 2 questions to the panel. We have one here, and the questions that we received online, we are transmitting directly to the speakers, and some of them are very specific, that I think that they're of much more of specific interest of one of the other. So thank you very much. Congratulations for the 3 speakers. And Paulo, I have 3 questions for you. One is the role of the meetings in these studies, the nucleus of technological information, and we showed that 56% of the startups were in São Paulo. And when you separate these 5 that reach the global level, how is this divided in São Paulo and the other states? And the other thing is the degree of investment that was done, and if you have this number, and I know that FAPESP's role is a key role compared to other states, and the level of participation of the students, especially of postdoc in these startups. This is a great question. The NITs NITs are they belong to the federal university, the public university, especially in the southeast region and in the south region of Brazil, and they participate in these partnerships with the startups. And regarding the specific values, we did not have this evidence, but it is an important evidence to be sought in future studies. And regarding the participation of students, yes, in some cases we were able to find a participation of students, but still very much reduced. So this would be a suggestion for the public policies, the involvement and the fostering of the researchers in this sense. Regarding these 5 startups, the great majority are in the state of São Paulo for sure. So, and they reach the highest levels of the study. So unfortunately, we have to close our panel.
colleagues for being with us online. And we will direct to you any further questions. I received some specific questions that I instructed the team to direct to you all. And I'm pretty sure that we will have some follow-ups from your presentations for all the three of you directly from my team here, at least. Thank you all for being with us. Muito obrigado aí a todos e a gente ainda tem mais uma sessão aqui. Obrigado.
We still have one session. Thank you very much. Thank you very much, Ricardo, for your moderation. Thank you, speakers, and thank you all. Well, moving on forward to their program, we invite as chair of our Promega panel, the Vice Director of Bio-Manguinhos, Marco Alberto Medeiros. He's a doctor in biotechnology by the Federal University of Pelotas. He has a postdoctorate in the University of California in Los Angeles and Pasteur Institute. And he acted as invited researcher in the vaccine developer in GSK lab in Vienna, Austria. And we also invite Promega Corporation senior researcher Juliano Alves, who will make the presentation platform of development of rapid and sensitive methods for analyte detection. He's a doctor in pharmacology by the Federal University of São Paulo. He was a researcher of the Genomic Institute for Novartis and also Script Research Institute. At Promega, he worked with the development of several technologies used in the breakthrough of medicines. The group leader in the Institute of l'École Normale Supérieure, Lionel Navarro, who will give his presentation, Plant and Microalgae Extracellular Vesicles as Carriers of RNA-based therapeutics. So we remind you all that this presentation has confidential classified data, so it will be interpreted for those who are online. For those who are present, we ask you that do not use your smartphones either in video or photos or any kind of recording. So he has experience in the study of interactions, pathogens, and hosts, and innate immunity, epigenetics, and silencing of RNA. His research team was pioneer in the role and the regulation of small intracellular RNAs in the interactions with gas hosts and bacteria, and the identification and characterizations of bacteria suppressors, and also in the transcriptional control of immunological for this methylation of DNA. So he founded several companies and spin-offs developing disruptive technologies to control infectious diseases of plants, animals, and humans. So he's currently director of research for Natural Recherche Scientifique in France. Scientifique. So I pass the floor to Marcos Alberto Medeiros.
Excuse me. I'd like to thank you, Lina and Julian, for accepting to talk in this event, the International Symposium. Yeah. I hope you enjoy your lecture. Go ahead, please.
Boa tarde a todos. Primeiramente, eu gostaria de agradecer.
You all, I would like to thank the opportunity of being able to talk to you all. My name is Juliano Alves. I am a senior researcher at Promega corporation, I'm going to talk about a set of solutions driven towards the immune messengers. And we talk about assays. We think about the ELISA assay, and we think, you know, that the time that is required to generate after the cleaning of the wells and the regulators, and obviously the time consumed with the proceedings are taken, since you take several hours to have the results. So thinking in a way of solving this process and to simplifying it, we thought about the Lumit platform. So can I change this? I'm not going to take the risk. So in this Lumit concept, these assays, to simplify, they consist in two primary antibodies that are combined with another substance, and they, in the presence of this analyte, they unite and they conduce to the complementation, the activation of these analytes. So in the substrate, you have the bioluminescent results. One of the advantage
you reduce, consequently, you reduce the time of incubation. And ELISA can lead several hours to obtain results, and this Lumit assay, the time is reduced from to 120 minutes, 2 hours. One of the advantages is the high sensitivity for those researchers that work with cells. There's a possibility of reducing the number of cells per study, and the assay window it's better, and also the development.
Do ensaio, você pode correr testes.
The possibility of we usually say that it's a homogeneous assay because it doesn't require the cleaning of the blades. You add reagents, you do the reading of the results, and it's easily adaptable to the heart. Once I've said that, we can envision a series of applications, possible applications for the Lumit platform, the detection, direct and indirect detection of analytes, protein-protein interaction studies, development of competition assays, and also the detection of antibodies. When it comes to the direct detection of analytes, the main requirement is that there should be two primary antibodies, and these antibodies should not be of different species. The advantage, as you maximize the number of antibodies that may be combined in order to select the pair that will be ideal for your assay in a short span of time. Using this approach, today we add many solutions, as you see on the screen, interferonogram and 2 and 4F alpha, as well as hereditary factors like VGF or insulin. But also there is a possibility of researchers generating their own in-house method through the use of assay kit, which has all the necessary components to conjugate your antibodies of interest, including fragments of the luminiferase. If the researcher does not want to generate his or her own reagents, the other option is to do the pairing of the antibodies of interest with secondary luminous antibodies. These antibodies, I can tell you, they come in different flavors, like anti-mouse, anti-rabbit, anti-goat, and anti-rat. The difference in that's adaptation is that you didn't have to do the conjugation, as I said before, and this is an approach that's used currently and intracellular events like phosphorylation and cellular degradation. Another possibility of this platform, as I said before, is that it allows for a study between protein to protein interactions with the use of antibodies that link to purification tags, such as this tag, plaque tag, or GST. This tool is used, for example, in screening of drugs like Protex that induce the proteolytic destruction of deregulated, unregulated proteins in cancer cases. Another possibility also is the development of competition assays in which a tracer molecule, be it a organic compound linked to a luciferase fragment or an antibody, for example, it's dislocated through the interest analyto losing luminescence, for example, bioluminescence. An example of the solution developed by Promega was this SCIN, in which you detect antibodies that link to the arsenal receptor. Now, this methodology can also be used in assays for the identification of metabolites. The last example I'd like to bring to your attention is the possibility of using this platform to detect platforms, be it for the confirmation of the exposure of individuals to certain pathogens, or to try to visualize the potential of neutralization of these antibodies in human samples. And we can use these tools also in the discovery of antibodies with therapeutics and antiviral activities. To exemplify what I just said, in the recent past, we've developed a limit biochemical assay that consists of the interaction. My pointer is not working too well, but I'll try to explain it to you better, step by step. We have developed an assay some time ago. It consists of the interaction between the RBD portion of the spike protein on SARS-CoV-2 with the soluble receptor. How does the method work? In the absence of an interference or an antibody or an organic molecule, these two proteins find themselves in a solution. They do protein-to-protein interaction, and these protections have an FC tail derived from an antibody, and that allows for the use of secondary movements antibodies that will lead to the reconstruction of analog luciferase and the generation of bioluminescence. The absence of an inhibitor agent, you have 100% luminescence. And when you have an antibody or a molecule or something that interferes in that interaction, the RBD will be sequestered and it will be kept from interacting with acid 2, and surely, consequently, antibodies won't be able to get close to the luminescence. The loss of bioluminescence in this case is a result of the presence of an antibody or a molecule that affects in that interaction. To demonstrate the concept and to prove the concept that this methodology works, we've done a limited screening to antibodies commercially available, and we can ascertain the fact that in the first part of the graph, and the samples where we didn't have any bars, these are antibodies against spike protein and SARS-CoV-2, and our method was able to detect these antibodies with a neutralizing potential, and the control antibodies didn't have any interference in the interaction between RBS2. It's worth highlighting that the bars in the middle of the first graph are cut in half. These are antibodies that were generated in the first outbreak of SARS-CoV in 2003, and they're able to interfere with up until a certain point. We have tested also sequences generated with in silico predictions based on the interface, the interface region between the acid2, S2, and RBD receptors, and we tested to see if we would be able to detect this interference. We confirmed the remarks published at that time that 3 sequences that the authors called mini proteins were able to interfere with the RBD S2 interaction. They demonstrate the potential of this platform to be used in discoveries of antibodies with therapeutic potential, in this case, antiviral activity. But it's not only that. You can do many studies, molecular studies. In this case, we looked into the role of the mutations present in RBD and the process of interaction with S2. Various mutations were reported on and many variants. And the concept is, depending on the affinity, probably you will need more analyto to be able to interfere in the RBDS2 interaction. To mutants that we have tested, we noticed that 3 mutations caused a dislocation of the curve, a displacement of the curve, which suggests that these mutations conferred a certain increase, a certain degree, an increase in affinity as for S2. Vis-a-vis S2. In addition, we looked into whether these mutations would have a role in the process of evasion of the immunological system, of the immune system. That is, if they would prevent the linkage of the antibody. And we had tested a small panel of antibodies. We went back to that panel. We generated RBDs with FC portions, so we could just replace them with the RBD that we have in the assay. And we noticed that for 2 antibodies that led to complete neutralization, 2 mutations were caused. They caused the complete loss of ligation. And it's worth noting that one of these mutations, N501Y, has a double role. It plays a double role. It apparently increases affinity with S2, but it also inhibits the linking of antibody that was previously able to neutralize the interaction between RBD and S2. And we looked into mutations of variants coming from sequences coming from variants of SARS-CoV-2 from the United Kingdom, South Africa, and Brazil. We noticed the same effect, since these 3 variants have that mutation, which reinforces the remark that this mutation can lead to an escape of the immune system. When it comes to human samples, we did an analysis of samples that were positive, confirmed for PCR, and negative samples, so as to observe the distribution of those samples in order to ascertain a cutoff that could help us when we would test unknown samples. When it came a time to test unknown samples, as we can see in both cases, third samples of serum and plasma, and we could notice that negative samples began to group together below 30% at a neutralization value of 30%. As for positive samples in green, we noticed that these samples, most of them tend to group together, to come together between 80% and 100%. With this information, we have defined a cutoff line and 30% neutralization so as to distinguish between positive and negative samples, and that led to a performance of a sensitivity of roughly 80% and a specificity of approximately 100% in these cases. With this criterion, the way we have defined it, we ran a temporal analysis of samples of patients that were infected by SARS-CoV, and we've tested several samples throughout time, and we noticed that the antibodies that were able to interfere in the RBD and S2, they began to surface and on the 12th day, and these individuals that were affected, the antibodies lasted up to 6 months. So similarly, we tested also our methodology with patients that were immunized with the coronavirus vaccine, and we noticed that only those individuals that were immunized after the 2nd dose, there was a spike in the immunization. The sample was able to completely eliminate the bioluminescence signal in our methodology. These results were published recently, and recently we used this information the lessons learned in the course of the pandemic to expand and try and develop new serological solutions using the GOMET platform, and two neglected diseases that we are keen on developing are Zika, because of the historical background, and dengue fever, and because that is a problem, a public health problem in Brazil and so many countries, and a concept of the assay, the design of the assay is a bit different, because in this case we used antigens related to nospherase fragments, and if there are antibodies present that recognize that antigen, that antibody during the linking process will complement nospherase, generating a bioluminescent signal. And preliminary results we can observe in the graph on the right side of the screen, on the left side of the screen, excuse me, several antibodies allowed that we were able to detect several of them, which makes us see that we can use that platform for other purposes. Different volumes of reactions can be used, showing the flexibility of this platform and miniaturizing and maximizing the use of the reagent. But we also know that serological assays for the detection of antibodies against arboviruses, there's a problem of cross contamination. We use polyclonal antibodies for dengue. Our kit was able to detect, that's the first graph. To mitigate this problem, we developed a blocking agent that was able to neutralize the presence of this antibody, contaminant antibody, but most importantly, the presence of this blocking reagent didn't interfere in the reaction and the identification of Zika antibodies. It's still in progress. And parallel to it, using the data from the same concept, we're developing a test to detect anti-dengue antibodies following the same principle, using antibodies antigens. And preliminary results we were able to detect. And in this case, the same problem surfaced when it comes to Zika, but we were able to develop a blocking agent in this case that inhibits a crossed reaction, but it doesn't interfere in the results. But it's still a project underway. It's currently underway, and we'll have more details to talk about it later on this topic. Just to wrap up my remarks, today I presented to you the LUMIT platform, the detection of the interaction between virus proteins and infected animals and their proteins. It's easy to then host proteins. This methodology can be used in addition to a virus neutralization platform, be it immunized animals or not. This molecular studies can be done through this platform to understand the role of mutations in the interaction between these two proteins, and also they can expand it to other viruses of interest. And most importantly, this assay can be automated and miniaturized. To wrap up, I'd like to acknowledge all the scientists involved in the study. We can't do anything alone. We depend always rely on a network of collaborators. And I would like to extend a warm thank you to all the participants of this panel. And now I'm available for Q&A. After Leonel's presentation, we'll run a series of questions after Leonel's presentation. Byron, please.
Do you have a pointer? Pointer. Oh, sorry. Congratulations, Liner and Julian, for nice talk. I think you have a time for a few questions because the time is almost finished for you. People need to go to lunch and back for the next session. The Paulo.
Zé Paulo, IOC, Fiocruz, CNPEM, parabenizar os dois palestrantes.
Congratulate both speakers. Julian, I would like you to draw a comparison between Lumit and Luminance, advantages and disadvantages, pros and cons of the platform that you presented related to luminance. That's a good question. Luminex is the assay consists of the use of beads for magnetics in which you're able to do the physical separation. In the case of Lumit, that's not necessary because you do the assay in solution. You don't have to do the physical separation of the analyto in your sample. That's one of the biggest pros, advantages of Luminex. But the difference of the Luminex and another difference between Luminex and Luminex, and I have to actually accept my drawbacks here, because the assay depends on the TF-RAT, which you might be looking at different wavelengths. And at this time with Luminex, that's not possible. But let's wait the next events and the next breakthroughs. That's a good question. Excuse me, but the time is up. Yeah.
The ladies come for, they say, finish.
Obrigada, senhores. Muito obrigada.
Thank you, gentlemen. Thank you very much. And now we'll have a break for lunch and to visit posters and the presentation of Fix and Lunchbox. I would like to invite all of you to visit the area of stands and the booths of exhibitors located in the external area. We'll be back in 1 hour and 30 minutes. Thank you.